Imagine stepping into a hyper-sterile, climate-controlled laboratory where the hum of automated liquid handlers replaces the traditional sounds of biological research. Here, beneath glowing monitors displaying complex molecular structures, scientists are actively constructing nature. Vials of clear liquids contain the fundamental chemical precursors of existence: adenine, cytosine, guanine, and thymine, waiting to be assembled into sequences that have never before existed. This is the frontier of synthetic biology. The profound stakes of this endeavor cannot be overstated. For billions of years, the architecture of life was dictated by the chaotic, unguided processes of natural selection. Now humanity stands at the precipice of a radical new era. By approaching biological systems as modular, programmable components, researchers are attempting to exert unprecedented control over the fundamental machinery of life. This promises scientific breakthroughs that could completely redefine our relationship with nature. To understand the magnitude of this transition, we must examine the core mechanism of DNA synthesis. For decades, the biological sciences were largely confined to observation, mapping genomes to understand what nature had already built. The pivotal shift from reading the genetic code to actively writing it gained immense momentum in the early 2000s as automated synthesizers became commercially viable, sequentially adding nucleotide bases to print artificial genetic material. This capability to print custom DNA sequences fundamentally alters our interaction with the biological universe. Instead of relying on existing templates, scientists computationally design a desired genetic sequence, transmit that data to a synthesizer, and receive a physical vial containing the engineered DNA. It is a monumental leap from passive biological observation to active biological authorship, transforming abstract digital data into tangible, functioning biological matter. The scientific consequences of this paradigm shift are reshaping the boundaries of engineering and research. We are moving beyond simply modifying existing organisms to a future where we design bespoke biological machines tailored for specific tasks. This transition elevates biology from a descriptive science to a prescriptive engineering discipline, characterized by standardization, abstraction, and highly predictable design cycles. By harnessing the efficiency of synthetic life, researchers are unlocking unprecedented possibilities for solving global crises. Engineered microbes could be deployed to break down persistent environmental pollutants, synthesize resilient crops capable of withstanding extreme climate shifts, or manufacture sustainable alternatives to petrochemical plastics. The profound potential of synthetic biology lies in its capacity to leverage the self-replicating nature of living systems to address challenges traditional engineering cannot solve. To conceptualize how these synthetic systems operate, leading researchers at the Massachusetts Institute of Technology have increasingly adopted a powerful metaphor: the code of life as software. In this framework, the cell is viewed as the hardware, the complex biochemical machinery that executes instructions, while the DNA sequence serves as the programmable software. Just as computer scientists write lines of code, synthetic biologists compile genetic sequences to instruct cellular machinery. This software metaphor extends deeply into the methodology of the field. Scientists utilize specialized computer-aided design programs to assemble genetic circuits, stringing together biological logic gates that allow cells to sense their environment and compute responses. If an engineered organism fails, researchers can sequence its genome to debug the biological code. By treating genetics as a programmable language, synthetic biology aims to make living systems as scalable as modern software. Yet this modern vision of programmable life did not emerge in a vacuum. It is the culmination of a historical continuum of genetic research stretching back over half a century. To truly grasp the revolutionary nature of contemporary synthetic biology, we must bridge the theoretical future back to the foundational steps that made it possible. The journey began in earnest during the early 1970s at Stanford University. These pioneering experiments marked the birth of genetic engineering, shattering the assumption that the genetic boundaries between species were immutable. By proving that DNA from different species could be successfully joined together in a laboratory setting, early researchers laid the critical groundwork for today's synthetic revolution. They established the fundamental premise that DNA is a universal language, capable of being read and executed by almost any living organism. The scientific principles underlying these early successes were rooted in recombinant DNA technology. This process relied heavily on the discovery of restriction enzymes, naturally occurring proteins found in bacteria that act as molecular scissors, capable of cutting DNA molecules at highly specific recognition sites. By using these enzymes to cut a desired gene and slice open a vector molecule, scientists created complementary ends that naturally bonded together. Once these separate pieces of genetic material were joined, an enzyme called DNA ligase was introduced to permanently seal the chemical bonds, creating a continuous strand of recombinant DNA. This engineered molecule could then be introduced into a host organism, which would read the foreign genetic instructions. This cut-and-paste methodology enabled the mass production of vital human therapeutics, such as synthetic insulin, within bacterial bioreactors. Despite these monumental early victories, the procedural reality of genetic engineering throughout the 1980s and 1990s was fraught with staggering limitations. The restriction enzymes utilized in early techniques were relatively blunt instruments, recognizing only short, specific sequences of DNA that occurred frequently and randomly. As documented by researchers modifying complex organisms, this lack of precision meant scientists had very little control over where a new gene would integrate. This unpredictable integration often resulted in catastrophic failure rates, as inserted genes could disrupt essential existing functions, trigger unwanted mutations, or simply fail to express themselves altogether. The process was agonizingly slow, requiring researchers to screen thousands of modified cells just to find a single viable specimen. For decades, this inherent friction and inaccuracy frustrated scientists seeking total biological control, making precise engineering seem agonizingly out of reach. The landscape of genetic manipulation was irrevocably transformed in 2012 when researchers at the University of California, Berkeley, published a landmark paper detailing a revolutionary molecular tool, the CRISPR-Cas9 gene editing system. Originally discovered as an ancient immune defense mechanism utilized by bacteria to fend off viral attacks, CRISPR was brilliantly repurposed into a highly programmable molecular scalpel, utilizing a customizable guide RNA molecule to direct a cutting enzyme. Unlike the blunt restriction enzymes of the past, the CRISPR-Cas9 system provides unprecedented precision, allowing researchers to target a specific sequence of DNA among the billions of base pairs that make up a complex genome. Once the guide RNA locates its exact genetic match, the Cas9 enzyme makes a precise cut. Scientists can then rely on the cell's natural repair machinery to insert an entirely new synthesized genetic payload. The widespread adoption of CRISPR drastically lowered the barriers to genetic research, fundamentally accelerating the biological timeline from decades of painstaking trial and error to mere months of targeted experimentation. Because designing a new guide RNA is incredibly cheap and straightforward compared to older editing methods, laboratories around the world rapidly integrated CRISPR into their workflows, democratizing access to advanced genetic engineering by 2015. This democratization of gene editing fundamentally altered institutional timelines and the pace of biological discovery. Projects that previously required millions of dollars could now be executed efficiently by small teams. More importantly, the immense reliability and precision of CRISPR finally provided the level of predictability that geneticists had long sought, effectively closing the chapter on rudimentary gene splicing and setting the definitive stage for true synthetic biology. While CRISPR revolutionized editing the code of life, it remained constrained by one reality. Researchers were modifying an existing biological template. To truly master biology, scientists needed to move beyond rewriting scattered sentences in a genetic manuscript. The ultimate frontier was de novo synthesis, the audacious ambition to write entirely new DNA sequences from scratch, completely free from the blueprints inherited from nature. This leap required a profound shift, transitioning from biological manipulation to pure chemical engineering. Instead of relying on living cells to copy genetic material, researchers developed machines capable of assembling raw chemical precursors into custom instructions. It was about conceptualizing a biological function on a computer, synthesizing the corresponding DNA, and breathing life into a novel entity. The physical mechanics of printing artificial DNA rely heavily on phosphoramidite chemistry. Inside modern synthesizers, the four essential building blocks of life, adenine, cytosine, guanine, and thymine, are stored as liquid precursors. Directed by a digital sequence file, automated valves deliver these nucleobases into a reaction chamber, chemically binding them to a solid support matrix in a controlled cycle. Because this chemical process becomes prone to errors as the molecular chain grows, machines synthesize short fragments of DNA, known as oligonucleotides, no more than a few hundred base pairs in length. To construct a complete genome, these short fragments must be stitched together using specialized enzymes, which recognize overlapping sequences and fuse them into a continuous double helix. Constructing massive genetic sequences depended on a radical shift in manufacturing economics, captured by the Carlson curve. Named after researcher Rob Carlson, this metric serves as the biological equivalent of Moore's law, tracking the exponential increase in the speed and capacity of DNA synthesis. As automation took hold, the efficiency of writing genetic code doubled at a staggering rate. In the early days of genetic engineering, synthesizing a single base pair of DNA was a slow process, costing tens of dollars. By the early 2000s, the Carlson curve dictated a precipitous drop in these costs, plunging the price of synthesis to fractions of a cent per base pair. This profound economic deflation made it financially feasible to contemplate synthesizing entire genomes. At the dawn of the 21st century, this convergence of chemical precision and plummeting costs ignited an intense race across the global scientific landscape. Research universities and private institutes mobilized their brightest minds to chase the holy grail of molecular biology, creating a fully synthetic organism. The atmosphere was electric, driven by a relentless desire to prove that the software of life could be authored by human hands. Within these pressurized environments, the hum of automated sequencers and the glow of computational models provided the backdrop for a profound philosophical inquiry. Scientists were no longer observing the natural world. They were actively attempting to engineer it from the ground up, forcing a rigorous examination of the definition of a living system. The quest demanded a holistic understanding of the interconnected metabolic pathways required to sustain a cell. At the epicenter of this monumental endeavor was the J. Craig Venter Institute, an organization renowned for its pivotal role in sequencing the human genome. Founded by the pioneering geneticist Craig Venter, the institute assembled a multidisciplinary team of molecular biologists, computer scientists, and chemists. Their singular objective was to push the boundaries of synthetic genomics further than any institution had dared to venture. The researchers walking the halls of the institute carried the extraordinary weight of knowing they were attempting to cross a threshold that would forever alter humanity's relationship with nature. There was a palpable sense of awe mixed with deep trepidation as they prepared to execute an experiment challenging beliefs about natural creation. Every experimental failure was magnified by the understanding that they were closing in on manufacturing life itself. The intellectual and reputational stakes for Craig Venter and his colleagues were staggering. A successful synthesis would cement their legacy, but failure would invite severe professional scrutiny. Furthermore, they were acutely aware that their work would immediately trigger intense ethical debates regarding biosecurity and the moral implications of their unprecedented power. To transform their grand ambition into reality, the team realized they could not simply replicate a complex, naturally occurring genome. The sheer volume of genetic code in even a simple bacterium presented an overwhelming probability of chemical errors during synthesis. Therefore, before building a new life form, they first had to answer a deceptively simple question. What was the absolute minimum amount of genetic information required to keep a biological machine alive? To determine this baseline, the researchers turned to Mycoplasma genitalium, a bacterium possessing one of the smallest known genomes in the natural world. Through a painstaking process of targeted mutagenesis, the team systematically knocked out individual genes, observing whether the modified bacterium could still survive and replicate. If the cell died, the gene was deemed essential. If the cell continued to divide, the gene was crossed off their blueprint. This exhaustive process of biological subtraction took years of rigorous testing, slowly whittling away the evolutionary baggage accumulated over billions of years. Ultimately, the team identified a core set of just under 400 essential genes, constituting the absolute bare minimum instruction set necessary for life. This stripped-down, highly optimized genetic blueprint provided the exact digital sequence needed to attempt the physical construction of a fully synthetic organism. With the theoretical map of the minimal genome securely stored as a digital text file, the project shifted from an exercise in genomic reduction to a monumental feat of physical engineering. The challenge was no longer determining what to build, but figuring out how to accurately manufacture millions of base pairs of continuous DNA without a single catastrophic error. The researchers had to translate their optimized computer code back into the messy reality of biological chemistry. This transition required scaling up their synthesis capabilities to unprecedented levels. The team ordered thousands of short, custom-printed DNA fragments from commercial synthesis companies, preparing to execute a complex assembly process that had never been attempted at such a massive scale. The stage was finally set for the most critical phase, stitching these fragmented chemical components into a single, cohesive, and functional biological operating system. In 2010, the team executed the final historic sequence of procedures. Because a complete genome is too fragile to be assembled in a standard chemical solution, researchers utilized the natural DNA repair mechanisms of living yeast cells to act as a biological factory. They introduced the thousands of printed DNA fragments into the yeast, where specialized cellular machinery seamlessly stitched the overlapping segments together, constructing the complete synthetic chromosome precisely according to the digital blueprint. Once the assembly was complete, the team extracted the artificial genome from the yeast and transplanted it into a hollowed-out recipient cell of a different bacterial species, one emptied of its native DNA. As the synthetic genetic code took control of the host cell's machinery, it began producing entirely new proteins, effectively overriding the biological identity of the recipient. This successful transplantation marked the birth of Mycoplasma laboratorium, the first self-replicating organism on Earth whose entire genetic heritage was dictated by a computer file. To understand the monumental nature of this 2010 achievement at the J. Craig Venter Institute, we must view the cell not just as a biological entity, but as a microscopic computer. When the researchers injected the synthesized chromosome into the hollowed-out recipient cell, they were essentially installing a new chemical operating system. This mechanism of genome transplantation acted as a biological boot-up sequence, initiating a cascade of reactions that brought the artificial code to life. The newly introduced synthetic genome immediately began reading its environment, overriding the native instructions that previously governed the host. By hijacking the cellular machinery, the digital blueprint forced the organism to synthesize entirely new proteins. In a matter of hours, the recipient cell was completely transformed, abandoning its original biological identity and beginning to replicate as an entirely new, human-designed species. This successful activation sent immediate shockwaves through the global scientific community. The publication of these findings in the journal Science provided undeniable validation that synthetic biology was no longer a theoretical pursuit, but a functional reality. Academic institutions and research laboratories worldwide recognized that a critical threshold had been crossed, marking the definitive transition from merely reading genetic sequences to actively writing them. The institutional consequences were immediate and far-reaching. Funding paradigms shifted overnight, and universities began establishing dedicated synthetic biology departments to capitalize on this breakthrough. The era of custom-built organisms had officially begun, signaling to researchers that the fundamental building blocks of life could now be engineered with the same deliberate intent as software or mechanical systems. To prove definitively that their creation was a synthetic original and not a natural contamination, the research team embedded an unmistakable human signature within the DNA. They experienced a deep sense of intellectual awe as they utilized the genetic alphabet of adenine, cytosine, guanine, and thymine to encode hidden messages directly into the living genome. This technique of genomic watermarking ensured that the organism carried a permanent, verifiable record of its artificial origins. Within the twisting double helix of this microscopic entity, the scientists spelled out the names of the 46 contributing researchers and a specific website address. More remarkably, they encoded famous literary quotes, including words from physicist Richard Feynman. "What I cannot create, I do not understand." This act of weaving human language into the chemical fabric of a living cell evoked a powerful sense of wonder, forever signing their biological masterpiece. Yet, while the creation of a single synthetic cell was a historic triumph, it was essentially a flawless replication of an existing natural genome. The narrative of synthetic biology quickly pivoted from this proof of concept toward a much broader and more demanding ambition. Scientists set their sights on designing entirely original, multi-gene biological circuits that did not exist anywhere in the natural world. Engineering these complex circuits required a fundamental shift in methodology. Researchers moved beyond simply ensuring a cell's basic survival and began programming specific, highly coordinated tasks. They needed to string together multiple genetic sequences to function like logic gates in a silicon microchip, a daunting challenge that demanded unprecedented precision in biological design. To manage this escalating complexity, the field embraced the concept of biobricks, an initiative heavily championed by researchers at the Massachusetts Institute of Technology. Biobricks are standardized genetic sequences, each with a specific, well-defined function, designed to be snapped together just like biological Lego bricks. This system forced the inherently fluid nature of biology into predictable, modular engineering components. By cataloging these standardized parts in an open-source registry, academic institutions created a universal language for biological construction. Scientists around the globe could now select a promoter, a coding sequence, and a terminator from a library of biobricks, assembling them to create novel genetic machines. This standardization was a critical step in transforming biology from a descriptive science into a rigorous engineering discipline. However, living systems actively and aggressively resist human engineering. As researchers deploy these standardized parts, they face the relentless challenge of biological noise, which manifests as random fluctuations in gene expression and unpredictable cellular behavior. The chaotic nature of living organisms means they constantly attempt to mutate, evolve, and often break the synthetic circuits placed inside them. To combat this biological noise, engineers implement meticulously documented procedures and structural safeguards. They design negative feedback loops to regulate gene expression and utilize genetic insulation to prevent synthetic pathways from interfering with the host cell's natural functions. Furthermore, they mathematically model the metabolic load, ensuring the artificial circuits do not exhaust the organism's energy reserves, which would prompt the cell to discard the engineered DNA entirely. Overcoming these chaotic unpredictabilities resulted in a monumental victory for engineering rigor over biological resistance. By successfully applying principles of control theory to cellular mechanisms, scientists achieved a new generation of stable, reliable synthetic organisms. These robust biological machines could maintain their programmed functions over countless generations without succumbing to evolutionary degradation. The achievement of these stable synthetic pathways transformed the field entirely. Engineered cells were now capable of executing highly complex, multi-step metabolic processes with the precision of a factory assembly line. This stability proved that synthetic biology could move beyond fragile laboratory curiosities, establishing a robust platform capable of sustained and predictable performance. With stable organisms finally achieved, the engineering focus expanded from microscopic petri dishes to massive industrial biomanufacturing. Industry white papers outline the immense thermodynamic and fluid dynamic challenges involved in scaling up synthetic biology. Moving a delicate cellular process from a controlled laboratory flask to a commercial fermentation tank holding hundreds of thousands of liters requires mastering entirely new physical forces. In these massive bioreactors, maintaining precise oxygen transfer rates, uniform temperature control, and even nutrient distribution becomes a monumental engineering feat. Fluid dynamic specialists must carefully calculate the shear stress exerted on the fragile cells by massive industrial impellers. Ensuring that synthetic microbes survive the violent agitation required for large-scale metabolism is essential to making biomanufacturing economically viable. Mastering this biomanufacturing scale-up serves as the critical bridge between theoretical science and tangible, world-altering applications. The foundational principles of genetic standardization, combined with the ability to cultivate engineered organisms by the ton, have paved the way for a new industrial revolution. The laboratory breakthroughs of the past decade are now ready to be deployed on a global scale. We now stand at the threshold of the application phase, where the abstract mastery of DNA translates into physical solutions for our most pressing global challenges. The narrative shifts from the intricate mechanics of how we build these living machines to the transformative reality of what they can produce. These microscopic factories are poised to rewrite the future of energy, materials, and human health. Step inside a cutting-edge biofuel facility, such as the Joint Bioenergy Institute in California, and traditional notions of manufacturing immediately shatter. There are no smokestacks belching soot. Instead, the space is dominated by the rhythmic hum of fluid pumps circulating liquid through towering photobioreactors. Inside these colossal vats, trillions of engineered algae are bathed in calibrated LED light, suspended in accelerated biological production. This is the visual reality of the synthetic biofuel industry, replacing fossilized sunlight extraction with real-time energy harvesting. Miles of surgical-grade steel piping connect these living vats, carefully regulating temperature and nutrient flow. The entire facility functions as a macrobiological organism, designed with one purpose: coaxing engineered cells into continuously excreting sustainable energy alternatives. Achieving this continuous production requires unprecedented metabolic engineering, effectively turning a single cell into a microscopic oil well. Scientists meticulously rewrite internal metabolic pathways, often starting with highly efficient strains of yeast. By overexpressing specific genes responsible for lipid production and deleting pathways that divert energy toward reproduction, researchers force the microorganism to funnel its metabolic energy into synthesizing hydrocarbons. The procedural mechanics of this biological hacking are extraordinary. Engineered microbes are designed to consume abundant, low-value inputs like agricultural waste or captured industrial carbon dioxide. Through synthetic pathways, they convert these inputs into energy-dense, drop-in biofuels. This is not theoretical. In 2018, the biotech company LanzaTech successfully fueled a commercial Virgin Atlantic flight using biojet fuel derived from waste gases fermented by engineered bacteria. Scaling these metabolic fuel excretors projects a massive economic and geopolitical shift, threatening the century-old petrochemical hegemony. For decades, global power dynamics have been dictated by geographic access to subterranean oil reserves, creating volatile supply chains. If high-performance fuels can be brewed in steel tanks anywhere on Earth using engineered microbes, the geographic monopoly on energy production evaporates. Beyond geopolitical disruption, the environmental consequences of replacing extracted fossil fuels are staggering. This technology offers a pathway to a closed-loop carbon economy. Because synthetic microbes consume carbon dioxide during their growth phase, the carbon emitted when the biofuel burns is effectively offset, creating a nearly zero-emission life cycle. We are witnessing the foundational steps toward decoupling human energy consumption from atmospheric greenhouse gas accumulation. This mastery over cellular machinery extends far beyond industrial energy, initiating a radical shift in human healthcare known as programmable medicine. Traditional pharmaceuticals operate as blunt chemical instruments, flooding the body with toxins to suppress symptoms. Synthetic biology, by contrast, allows scientists to design living, intelligent therapeutics that actively navigate the human body, assess cellular environments, and execute highly specific commands. The scientific logic behind programmable medicines relies on treating the human immune system as a customizable hardware platform. The most prominent example is CAR T-cell therapy, where a patient's own T-cells are extracted and genetically reprogrammed. Scientists insert a synthetic gene producing a chimeric antigen receptor, installing a biological targeting system that trains the cell to destroy specific cancers while leaving healthy tissue untouched. The speed at which synthetic therapeutics can now be developed was dramatically proven during the rapid response to the SARS-CoV-2 pandemic in early 2020. The moment researchers uploaded the genetic sequence of the novel pathogen to a global database, scientists halfway across the world downloaded the digital code. Without needing a physical sample, researchers used DNA synthesizers to print the required genetic instructions. This digital-to-physical pipeline fundamentally rewrote the procedural timeline of vaccine development. By utilizing synthetic mRNA encapsulated in lipid nanoparticles, scientists delivered these printed instructions directly into human cells, temporarily turning the patient's body into a vaccine manufacturing facility. What historically required years of isolating and cultivating live viruses was achieved in days through rapid digital prototyping and synthetic genetic assembly. The human stakes of this shift from chemical treatments to living drugs are perhaps the most awe-inspiring aspect of the synthetic biology revolution. Grounding this high-tech science in reality requires looking at patients pulled back from terminal diagnoses. In 2012, a young patient named Emily Whitehead became the first child to receive experimental CAR T-cell therapy for acute lymphoblastic leukemia, a disease resisting all conventional chemotherapy. Infused with her own genetically reprogrammed cells, Emily experienced a fierce biological battle as the synthetic T-cells multiplied, hunted down the leukemia, and eradicated it entirely. Today, her sustained remission stands as a testament to personalized cellular therapies. These living drugs outsmart previously incurable conditions by upgrading the patient's biological software, leaving behind a standing army of engineered sentinel cells to prevent relapse. Just as synthetic biology is reprogramming the human body, it is poised to reconstruct the physical world around us, giving rise to the factories of the future. Imagine walking into a visionary manufacturing environment devoid of the harsh chemical solvents of traditional industry. Instead, rows of precisely monitored fermentation tanks actively grow advanced biomaterials, secreting substances with physical properties that rival anything forged in a steel mill. In these biofactories, companies like Japan's Spiber have successfully engineered microorganisms to produce synthetic spider silk, brewing a material pound for pound stronger than steel. Meanwhile, researchers at Delft University of Technology have pioneered self-healing concrete by embedding dormant, limestone-producing bacteria into the building material. When a crack forms, the engineered microbes awaken, consume a packaged food source, and excrete calcite to physically seal the damage. The molecular logic driving these cellular factories relies on hijacking the most sophisticated manufacturing equipment in the known universe, the biological ribosome. Traditional chemical synthesis struggles to accurately assemble massive polymer chains, often resulting in toxic byproducts. Synthetic biologists bypass this limitation by writing custom DNA sequences that instruct the cellular machinery of yeast or bacteria to stitch together specific amino acids in a flawless, predetermined order. By reprogramming this microscopic machinery, scientists can dictate the exact length, structure, and sequence of the resulting proteins, achieving molecular precision that industrial chemistry simply cannot replicate. The engineered microbes act as programmable 3D printers operating at the nanometer scale, tirelessly assembling complex structural proteins and biodegradable plastics. Once the fermentation process is complete, the biological material is harvested, purified, and spun into highly advanced fibers. The successful deployment of these grown materials signals the dawn of a post-scarcity manufacturing paradigm, fundamentally altering global economics. For centuries, human industry has been defined by the extraction of finite resources, leading to fragile, planet-spanning supply chains. The bioeconomy promises a radical alternative: the ability to manufacture endlessly renewable high-performance materials using little more than engineered microbes, water, sugar, and basic elemental inputs. When the essential building blocks of civilization can be brewed locally in automated bioreactors, the very concept of resource scarcity begins to unravel. Cities could grow their own construction materials, textiles, and medicines on demand, drastically reducing the need to ship raw goods across oceans. This paradigm-shifting potential offers a tantalizing vision of a sustainable future where manufacturing capacity is limited only by the genetic software we can write. Beyond manufacturing, the most ambitious application of synthetic biology lies in planetary repair. Researchers are engineering specialized microbes designed to act as microscopic environmental janitors, capable of actively reversing ecological damage. Building upon the 2016 discovery of a naturally occurring bacterium in Japan that degrades plastic, synthetic biologists are now writing entirely new genetic sequences to hypercharge this process. These engineered organisms are being optimized to consume ocean microplastics at unprecedented speeds, breaking down resilient synthetic polymers into harmless organic byproducts. This concept of synthetic bioremediation extends far beyond plastic degradation. In advanced laboratories, scientists are designing synthetic cyanobacteria engineered to sequester atmospheric carbon dioxide at rates vastly exceeding those of natural plants. Other engineered strains are being programmed to neutralize toxic chemical spills, metabolizing heavy metals and petrochemical pollutants into inert substances. By deploying these living microscopic machines, we could theoretically launch a self-replicating, self-sustaining fleet of environmental scrubbers, offering a radical biological solution to the most pressing climate challenges of the 21st century. Yet the very power that makes these scientific breakthroughs so promising also casts a long, unsettling shadow. As we move from observing the natural world to actively redesigning it, we cross a threshold fraught with profound ethical dilemmas. The utopian vision of a healed biosphere and endless biological manufacturing is inextricably linked to the existential risks of unleashing artificial life forms. This transition marks a critical pivot in human history, shifting our technological focus from inert machinery to self-replicating biological systems. The tools of DNA synthesis that allow us to engineer life-saving medicines and environmental scrubbers are inherently dual-use. The same genetic engineering principles that empower us to cure diseases could, if misdirected, be used to engineer unprecedented biosecurity threats, forcing society to confront the terrifying reality of our new capabilities. At the heart of this technological revolution lies a deep philosophical unease often characterized as the dilemma of playing God. For millennia, humanity has selectively bred plants and animals, but creating life from scratch, molecule by molecule, represents a fundamental rupture with the past. When researchers led by Craig Venter successfully booted up the synthetic genome of Mycoplasma laboratorium, it was not merely a technical triumph, it was a philosophical earthquake that challenged our most basic definitions of what constitutes a living being. We are no longer simply reading the evolutionary history written in genetic code. We are authoring entirely new chapters that have no precedent in nature. This godlike capability stirs a primal apprehension. It forces bioethicists, theologians, and the public to grapple with the arrogance of assuming we can perfectly control the architecture of life. The realization that human hands can design, assemble, and animate a living organism from inert chemicals strips away the mystical veil of biology, leaving us with an awe-inspiring, yet deeply terrifying, responsibility. As artificial life moves from philosophical debate to commercial reality, it collides with the rigid structures of global commerce, triggering a massive evolution in intellectual property law. The legal foundation for this commodification of biology was laid in 1980 with the landmark United States Supreme Court case Diamond versus Chakrabarty, which ruled that a genetically modified oil-eating bacterium could be patented. The court famously declared that anything under the sun that is made by man is eligible for patent protection, setting a vast legal precedent. Today, this legal framework is being stretched to its limits by the advent of full genome synthesis. Biotech corporations are no longer just patenting minor genetic tweaks, they are claiming ownership over entirely novel synthetic genomes and the customized metabolic pathways within them. Intellectual property law is actively adapting to treat these engineered biological systems as proprietary software, transforming living, reproducing organisms into corporate assets, protected by the same legal mechanisms used to patent algorithms and industrial machinery. The bureaucratic reality of claiming ownership over the code of life is a meticulously documented process. When a biotech firm engineers a new organism using CRISPR or advanced DNA synthesis, their legal teams file exhaustive applications with the United States Patent and Trademark Office. These filings detail the exact sequences of artificial base pairs, the specific metabolic functions they encode, and the precise environmental triggers required to activate the engineered traits. This procedural machinery requires scientists to prove that their synthetic organism is entirely distinct from anything found in nature. Patent examiners review thousands of pages of genetic code, comparing the submitted sequences against global biological databases to verify their novelty. Once approved, the patent grants the corporation exclusive rights to manufacture, use, and license that specific biological entity, effectively placing a legal boundary around a newly invented species and strictly controlling who is allowed to reproduce it. The societal impact of allowing corporations to monopolize foundational biological technologies is a subject of intense, growing alarm. If the synthetic microbes required to produce next-generation medicines, drought-resistant crops, and sustainable biofuels are locked behind corporate patents, it threatens to create unprecedented global inequities. A handful of powerful biotech conglomerates could effectively control the essential biological infrastructure of tomorrow, dictating access to the very tools needed to ensure human survival in a changing climate. This aggressive patenting strategy risks stifling the broader scientific innovation it is meant to protect. When fundamental genetic circuits and synthetic pathways are heavily restricted, independent researchers and smaller laboratories are priced out of the field, unable to afford the exorbitant licensing fees. The long-term consequence is a monopolized biosphere, where the future of biological engineering is dictated not by public need, but by the profit margins of a select few corporate entities holding the keys to the genetic kingdom. This privatization of fundamental biology has ignited fierce ethical friction among independent researchers and advocates for open science. Pioneers in the field, including those involved in the BioBricks Foundation, have fought back by creating open-source registries of standard biological parts, arguing that the foundational code of life belongs to humanity as a whole. There is a palpable sense of injustice at the thought of corporate entities claiming exclusive dominion over the fundamental building blocks of existence. The thought that a corporation could own the exclusive rights to a synthetic organism capable of curing a global pandemic or cleaning the oceans strikes many as a deep moral failure. It transforms the awe-inspiring miracle of genetic engineering into a cold, transactional commodity. This tension between public trust and private ownership forces us to ask whether the immense power to write new biological code should be treated as a proprietary secret or a shared human inheritance. Yet, the debates over patents and laboratory ethics pale in comparison to the challenges that arise when these engineered organisms leave the controlled environment of the petri dish. The true test of synthetic biology is not whether an artificial cell functions perfectly within a sterile bioreactor, but how it behaves when introduced into the chaotic, infinitely complex web of the natural world. Transitioning from the laboratory to the wild requires an entirely different scale of risk assessment. In the lab, variables are tightly controlled, and a failed experiment can simply be sterilized and discarded. In the wild, there is no isolation chamber. The release of a synthetic organism introduces an unpredictable, self-replicating agent into an environment shaped by billions of years of intricate evolutionary balance. Consider the fragile complexity of a coastal estuary, a delicate natural ecosystem teeming with interdependent life forms. Here, microscopic phytoplankton form the base of a food web that supports everything from filter-feeding bivalves to apex predators. The soil and water are governed by a precise chemical balance, maintained by millions of naturally evolved microbial species, interacting in ways that modern science still struggles to fully map or comprehend. Ecological modeling reveals the terrifying vulnerability of such environments to novel biological intrusions. If a synthetic microbe, designed to aggressively consume organic waste, were introduced into this estuary, the initial environmental cleanup could rapidly spiral out of control. The engineered organism might outcompete native bacteria for resources, triggering an unforeseen ecological cascade that collapses the local food web, vividly illustrating the devastating potential of inserting artificial life into the delicate machinery of nature. To understand the true scale of ecological intervention, we must look beyond single organisms and examine technologies designed to alter entire species. In classical genetics, a trait has a 50% chance of being passed from parent to offspring. A CRISPR-based gene drive shatters this fundamental biological law by utilizing a mechanism that actively copies and pastes itself onto the corresponding chromosome of a reproductive partner. This autonomous copying mechanism guarantees that nearly 100% of offspring inherit the engineered trait, bypassing natural selection entirely. Instead of waiting for a beneficial mutation to slowly propagate over millennia, a synthetic gene drive forces a specific genetic edit rapidly through a wild population, turning the slow march of evolution into a high-speed, directed sprint. The mechanics of this genetic override are no longer purely theoretical. In 2018, researchers at Imperial College London successfully demonstrated a gene drive designed to crash populations of the primary mosquito vector for malaria in sub-Saharan Africa. The laboratory procedure involved micro-injecting mosquito embryos with a payload targeting a highly conserved gene responsible for female fertility. Once these engineered insects matured and mated with wild-type mosquitoes, the synthetic sequence activated. Mathematical models published by the research team traced the exponential spread of the sterility trait across generations. Within a controlled environment, the gene drive reached total prevalence in just 7 to 11 generations, causing the confined mosquito population to collapse completely in less than a year. The permanent release of such an unstoppable genetic mechanism carries monumental risks that extend far beyond the target species. A 2016 report by the National Academies of Sciences, Engineering, and Medicine warned that an unchecked gene drive could trigger irreversible ecosystem alterations. If a drive designed to eliminate a pest jumps to a closely related, ecologically vital insect, the results could be catastrophic. This localized extinction would act as a domino effect, potentially initiating a vast food web collapse within the affected biome. Furthermore, an imperfectly copied gene drive could lose its intended function while retaining its aggressive inheritance mechanism, spreading an unpredictable genetic flaw through a global species with no known method of recall or reversal. Recognizing these immense hazards, synthetic biologists have spent the last decade architecting sophisticated biological containment systems. The goal is to build fail-safes directly into the genetic architecture of an engineered cell, ensuring it cannot survive outside a highly monitored laboratory. One primary strategy is synthetic auxotrophy, where researchers rewrite an organism's metabolism so that it absolutely requires an artificial nutrient to survive. Without a constant supply of this synthetic amino acid, the organism's internal machinery grinds to a halt. In tandem with auxotrophy, scientists construct genetic kill switches. These are intricate molecular circuits programmed to trigger cellular suicide if the microbe detects environmental changes, effectively dissolving the synthetic life form before it can reproduce in the wild. Validating these genetic kill switches requires rigorous adversarial testing to ensure flawless execution under stress. In 2015, researchers at the Massachusetts Institute of Technology designed a suite of biocontainment circuits in bacteria and subjected them to extreme evolutionary pressure. The laboratory protocol involved starving the microbes, bombarding them with mutagens, and placing them in environments mimicking natural soil and water. Technicians monitored the stress-tested populations for weeks, searching for any signs of escape or survival. The results demonstrated that the engineered failsafes activated as programmed, cleaving the bacterial DNA into harmless fragments the moment the survival parameters were breached. The researchers achieved an escape rate of less than 1 in 100 billion cells, setting a new benchmark for stability. Yet, empirical data from evolutionary biology suggests that absolute control over a replicating entity may be an illusion. A 2020 review published in the journal Nature Communications analyzed the long-term efficacy of genetic firewalls, concluding that life possesses an inherent, relentless drive to bypass constraints through constant natural mutation. Over billions of generations, even a microscopic escape rate provides enough statistical opportunity for a random mutation to deactivate a kill switch. If a vast industrial facility utilizes trillions of synthetic microbes, that infinitesimal margin of error translates into millions of potential escapees. Once a single mutated cell adapts to survive without its synthetic tether, the biological firewall is permanently breached. While the accidental release of a self-sustaining synthetic organism presents a formidable ecological challenge, the field faces an even darker vulnerability. The same tools used to engineer sustainable fuels or eradicate disease can be deliberately inverted. In 2017, the United States Department of Defense commissioned the Jason Advisory Group to evaluate this shifting landscape of genetic engineering. Their findings marked a stark pivot in global security protocols. The report underscored that as the foundational principles of synthetic biology become standardized and widely understood, the barrier to entry for creating weaponized biological agents drops precipitously, opening the door to malicious actors intent on engineering targeted harm. This escalating threat is deeply intertwined with the rapid democratization of biological creation. Today, the infrastructure required to manipulate genetic code is no longer confined to highly secure pharmaceutical laboratories. In modern community spaces from Brooklyn to Berlin, independent biohackers can access open-source genetic databases and automated pipetting robots for a fraction of their historical cost. The most critical shift in this decentralized environment is the advent of desktop DNA synthesizers. These compact machines can assemble custom genetic sequences on demand. A user simply downloads a digital blueprint from an online repository, feeds chemical building blocks into the device, and waits as the machine physically prints the genetic code, dispersing the tools of creation far beyond traditional regulatory oversight. The foundation of this decentralized power relies on an expansive architecture of global genetic data sharing. Platforms maintain publicly accessible archives containing the complete genomic sequences of millions of organisms, including highly lethal human pathogens. According to a 2022 Nuclear Threat Initiative white paper, this open science model, while crucial for medical advancement, creates a massive vulnerability in international biosecurity. An individual with malicious intent no longer needs to physically acquire a dangerous virus from a secure natural reservoir. Instead, they can simply download the pathogen's genetic blueprint, divide the sequence into smaller, inconspicuous segments, and order those fragments from commercial DNA synthesis providers. This digital-to-biological pipeline effectively turns data itself into a severe and immediate biosecurity hazard. The democratization of genetic data introduces a complex challenge known as dual-use research of concern, or DURC. This classification applies to life sciences research intended to yield clear benefits that could be misapplied to cause harm. Modern scientific publishing routinely uploads methodologies to global databases, inadvertently providing a blueprint to weaponize biological agents. A stark historical instance occurred in 2001 when Australian researchers inadvertently created a highly lethal strain of mousepox while developing a viral contraceptive. By inserting a specific gene, they unintentionally suppressed the mice's immune response. Publishing these modifications revealed that the same synthetic pathways could be adapted for human viruses, effectively distributing a recipe for biological warfare. The consequence is the looming threat of engineered pathogens designed to evade known medical countermeasures. In a 2018 report at the Johns Hopkins Center for Health Security, experts modeled a synthetic respiratory virus deliberately engineered to resist existing therapies. This simulation demonstrated that a novel pathogen could overwhelm global healthcare infrastructure within weeks. Unlike natural outbreaks, an engineered pandemic would lack a natural ecological footprint. Malicious actors could splice together the transmission efficiency of one virus with the lethality of another. This transforms the theoretical risk of a laboratory leak into an active, asymmetrical threat, allowing a small group to synthesize a catastrophic biological event from scratch. To counter this unprecedented risk, a global institutional defense network has been mobilized. The strategic logic relies on establishing international biosecurity screening protocols that monitor the flow of genetic material. Organizations like the International Gene Synthesis Consortium now require member companies to screen incoming orders against databases of regulated pathogens. When a researcher requests a specific sequence, specialized algorithms compare the digital code against the genomes of dangerous toxins. If a match is detected, the system flags the order for human review, forcing the vendor to verify the customer's identity. This ensures the physical production of synthetic DNA remains securely gatekept. These digital checkpoints represent a permanent transformation in how biological research is conducted. Every commercial request for custom genetic material now leaves a digital footprint, establishing a global surveillance infrastructure designed to catch malicious orders before they are physically printed. Consequently, using advanced gene editing tools like CRISPR is no longer an isolated laboratory activity, but a monitored transaction. Institutional review boards and national intelligence agencies increasingly collaborate to audit these synthesis records, ensuring the tools used to rewrite life are not diverted toward catastrophic ends. Yet, severe limitations in global biosecurity enforcement remain, exposing dangerous gaps in this defensive net. Because screening compliance is largely voluntary and concentrated among major industry players, a shadow market of unregulated synthesis providers operates outside the purview of international consortiums. The proliferation of decentralized benchtop DNA printers further erodes this institutional control. As the physical hardware required to print genetic code becomes cheaper, independent operators can bypass commercial screening altogether, rendering centralized checkpoints obsolete. The psychological weight of this biosecurity threat places an unprecedented burden on the technicians working on the front lines of genetic engineering. Laboratory workers dedicated to curing genetic diseases now operate under the sobering realization that their daily innovations could engineer the next global pandemic. For communities near high-containment synthetic biology facilities, the abstract fear of an engineered pathogen translates into tangible anxiety regarding municipal emergency protocols. The promise of miraculous medical advancements is permanently entangled with the dread that a single lapse in oversight could unleash an irreversible crisis. Moving beyond immediate security concerns, these capabilities force a deep philosophical reckoning regarding the fundamental nature of existence. In 2010, the J. Craig Venter Institute catalyzed this shift by announcing the creation of Mycoplasma Laboratorium, the world's first synthetic cell governed entirely by a human-designed, computer-generated genome. This landmark achievement shifted the scientific paradigm from editing existing organisms to engineering life from inanimate chemical precursors. By demonstrating that a living cell could be booted up from a digital file, the experiment fundamentally altered our assumption of life as an unbroken, naturally evolving lineage. Synthetic biology permanently dismantles traditional definitions of living things, blurring the boundary between biological organisms and engineered machines. The evolving scientific definition of life must now account for entities that possess DNA, consume energy, and reproduce, yet have no evolutionary ancestors. This architectural shift leaves one profound, unresolved question that the public record cannot fully settle. If an organism is built entirely from synthetic components to execute an industrial function, does it qualify as a living creature deserving of ethical consideration, or is it merely biological hardware? Even as scientific breakthroughs accelerate, the legal frameworks required to classify these novel entities remain fiercely debated. In response to this existential ambiguity, humanity is engaged in the active process of attempting to write the rules for its own evolution. The establishment of global bioethical guidelines has become a critical priority, resulting in summits where geneticists, ethicists, and policymakers draft frameworks to govern synthetic organisms. The durable consequence of this effort is the emergence of a new legal discipline dedicated exclusively to synthetic life, mandating rigorous ethical reviews before novel organisms are released. As we actively design the future of biology, these institutional guardrails represent our collective attempt to ensure our mastery over the genetic code elevates human civilization. The burden of these institutional guardrails falls heavily on tomorrow's bioengineers. For researchers working on DNA synthesis, the legacy of early milestones like the creation of Mycoplasma laboratorium by J. Craig Venter is not just a scientific triumph, but a mandate for caution. This shift requires every geneticist to act as a steward of the biosphere, weighing the profound societal responsibility of biological engineering against the drive for innovation. Their daily reality now involves navigating strict ethical review boards and constant biosecurity monitoring. Beyond the laboratory walls, we can envision a landscape transformed by these breakthroughs. Picture an agricultural zone where synthetic microbes, engineered using advanced CRISPR techniques, continuously monitor soil chemistry and synthesize targeted nutrients on demand. In this integrated future, the rigid boundary between human infrastructure and the natural ecosystem dissolves. Cities could seamlessly incorporate living materials, creating a synthetic ecosystem where engineered organisms actively scrub atmospheric carbon and metabolize urban waste. As we master these mechanisms on Earth, our gaze turns outward, expanding the scope of synthetic biology to the colonization of the universe. Astrobiologists are modeling how extremophile bacteria could be redesigned to survive the harsh radiation of the Martian surface. This interplanetary ambition introduces an unresolved question that current data cannot settle. Do we have the right to introduce synthetic microbes to alien environments, potentially destroying undiscovered biological signatures? Terraforming forces us to confront whether spreading terrestrial life supersedes preserving untouched worlds. Ultimately, the overarching consequence of synthetic biology is humanity's permanent transition from passive observers of natural selection to the active architects of evolution. This paradigm shift is cementing itself in international law, with global treaties actively classifying and monitoring synthetic genetic constructs. To manage this unprecedented power, regulatory bodies like the World Health Organization are now drafting frameworks for a global genomic registry. This database will permanently catalog synthetic lineages, ensuring that as engineered organisms integrate into our ecosystems, their artificial origins remain entirely traceable across future generations. For the agricultural communities on the front lines of this shift, the physical landscape is already transforming. Farmers no longer just harvest traditional crops. They cultivate living bioreactors, vast fields of engineered flora designed to actively sequester industrial carbon, fundamentally rewriting humanity's daily interaction with the natural world. When we peer through a microscope at a single synthetic cell dividing, we are no longer just observing the ancient machinery of nature. We are watching our own authored code boot up, a silent, microscopic testament to the exact moment humanity learned to write the language of life.